Hello my fellow pre-meds, and welcome back to our second video on ribonucleic acid. So in the first video we talked about transcription and the genetic code. In this video we're going to focus on translation and the regulation of genes. This is a dense topic, and if you haven't watched the previous video yet, I'd highly recommend it, as these concepts build on that video. Did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide deck. Links in the description down below, as well as the links for everything else. So let us get started over here. We're going to start with a light discussion on the structure and the different components of ribosomes. So ribosomes are ribonucleoproteins. That just means that they're a mixture of RNA, specifically ribosomal RNA, and proteins, and they carry out translation. The small ribosomal subunit binds mRNA, whereas the large subunit is responsible for forming that peptide bond, so the catalytic activity. Ribosomes contain tRNA binding sites, 3 tRNA binding sites specifically. So they contain the amino acyl, or the A site, the peptidyl, or the P site, and finally the exit, or the E site. Now, more on this in the next slide, and we'll clarify these with a diagram. Eukaryotic ribosomes are formed from a 40S and a 60S subunit. So the 40S is the small one, and the 60S is obviously the large one. And when they combine, they form the 80S ribosome, whereas prokaryotic ribosomes are formed from the 30S and 50S, and when they combine, they form the 70S ribosome. Now, why is 40S plus 60S 80S? Well, it turns out biochemists aren't the best mathematicians. I'm only kidding. The 40S and 60S is not like measuring mass. Like, it's not like you're saying 40 grams plus 60 grams is going to equal 100 grams. This is actually sedimentation rates, and it's determined experimentally. So when you experiment with the small subunit, you get 40S for the sedimentation rate. With the large subunit, you get 60, but when you combine them together, you get 80S. So it's not additive over there. Okay, so this is the structure of the ribosome. I don't want you to get distracted with all of the components of this diagram. I want you to focus on the ribosome itself. So you can see here's the large subunit, which is interacting with the tRNAs to form the peptide bond. More on that in a bit. And then the small subunit over here, which is interacting with the messenger RNA. And then I really want to focus on these 3 sites. So you have your amino acyl site, your peptidyl site, and your exit site. So translation will begin at the AUG start codon. So memorize that that is the start codon. However, in order for translation to occur, you first need to join the 2 subunits, the 2 ribosomal subunits with one another, because initially in the cell, they're dissociated, so they're each separate from one another. What's going to happen is once you have the small subunit bind the messenger RNA strand, then the first tRNA, which is carrying methionine, will join these two components, the messenger RNA and the small subunit. Once these 3 things come together, then and only then will the large subunit join and form the full ribosome. All right, so translation will proceed from the 5' to the 3' direction. So as you can see right here, the ribosome is going to move in this direction. So contrast this with DNA polymerase and RNA polymerase in the process of replication and transcription respectively. They proceeded from the 3' to 5'. In this case, it's from the 5' to 3'. All right, so now let's move on to the elongation of translation, the elongation phase of translation, which is a bit more complex, so bear with me. So these initial smaller diagrams are just what we saw in the previous slide. Now this is going to be the beginning of the elongation phase. So you have charred tRNAs are going to join at the A site. So in the previous slide we saw over here that the tRNA that's carrying methionine is going to join at the P site because it's the very first tRNA over there. So now it's at the P site over here. And then you're going to get the new tRNA, which let's say just it's carrying alanine, for example, is going to be at the A site. All right, so then what's going to happen is that the peptide bond is going to form at that point because you have 2 amino acids. So the peptide bond is going to form between methionine and alanine in this case. And then that cycle is going to continue to repeat until you start to see this long polypeptide chain. So it's repeated like 10, 20, or even 30 times, so you get this long growing polypeptide chain. Now notice this point over here, and I really want you to pay attention because this is a bit confusing. So the polypeptide chain, the growing polypeptide chain, is constantly being transferred between the peptidyl and the aminoacyl site. So if you take a look over here, you can see that right here, this is the growing polypeptide chain. It is on the tRNA that's on the peptidyl site. This new tRNA is charged with some amino acid. Again, let's just say it's alanine. It's at the aminoacyl site. And this is the exact moment that the peptide bond is forming. Now, once the peptide bond forms, the entire growing polypeptide chain moves from the tRNA on the peptidyl site to the tRNA on the aminoacyl site. So essentially, if you were to look at this, the split second after this occurred, you're going to see the growing polypeptide chain on the aminoacyl site. And then you have the tRNA that does not contain any amino acids on the peptidyl site. But at that instant, what's going to happen is you're going to have the process of translocation. What does this mean? That the ribosome is going to move 3 codons down the messenger RNA to move on to the next codon. When that happens, this is essentially what we have. When that happens, now the empty tRNA is on the exit site. The one with the growing polypeptide chain, which was initially on the aminoacyl site, is now at the peptidyl site. And now you get the new tRNA that's charged with a new amino acid on this A site. And what's going to happen now is that the one on the E site is going to exit, and you're going to repeat the process. I did want to clarify over here that there are 2 GTPs being consumed in the process of elongation during translation. These GTPs are being consumed, so one of them is going to be consumed when you have translocation. So when the ribosome moves along the mRNA strand, you need to use GTP to be able to mobilize it. The second one, a lot of students get it confused with the formation of the peptide bond, because it makes sense. If you're forming a bond, you need energy. But that's actually not when it's used. It's used when the new tRNA, when a new tRNA that's charged interacts with this A site. That is when you have the second GTP being consumed. Because when you're forming a peptide bond, it's true that you need energy, but recall from our previous slide deck, if you haven't watched it, definitely go check it out before watching this slide deck. But in the previous slide deck, we talked about how you form the interaction between the tRNA. So you have a tRNA that does not contain an amino acid, and then you get your amino acid, and using an enzyme called aminoacyl synthetase, you use the energy from ATP to form a high energy bond. And it turns out that high energy bond, we're going to harness that energy from that high energy bond to form the peptide bond. So that's why you don't need additional GTP molecules to form the peptide bond. The last step of translation is termination. So termination occurs when you have a stop codon. So essentially when you run into a stop codon, none of the tRNAs recognize that stop codon. So it's going to allow release factors to recognize it and terminate the translation. These stop codons include, let me put my pointer over here. So the stop codons include UAA, UAG, and UGA. You should memorize these. A mnemonic that I gave in the previous slide deck is you are annoying, you are gone, you go away. That's just how I memorize them. And then at the very end, the ribosomal subunits will dissociate from one another. So again, when they're in the cell outside of the process of translation, they're not joined to one another. They only join when translation occurs, and they're not degraded, so you can reuse them. Okay, now let's talk about post-translational processing. There's a lot over here, but to be honest, the most important point is that you have what's called phosphorylation, because this shows up with certain amino acids. If you recall from our amino acid slide deck, we talked about amino acids such as tyrosine, threonine, and serine, which contain an hydroxyl group in their side chain. These can be phosphorylated. So that's just post-translational processing that occurs. That's the most high yield part, but I'll also mention these because it's good to be familiar with these concepts. So the newly synthesized polypeptide must fold into its 3-dimensional structure. So this circles us back to the hierarchy, the structural hierarchy of proteins, which we have a slide deck on, we have a video on earlier in this playlist. So essentially when you have your polypeptide, it will then fold into the final 3D conformation. Chaperone proteins assist in this folding process. And then other modifications other than phosphorylation, glycosylation sometimes is something that shows up. Proteins are then transported out of whether they're going to be transported out of the cell or carry out their function within the cell. And then a lot of these post-translational processing activities occur to regulate the protein's activity, the stability, and like regulate enzymatic activity, for example, and the location of it. So that's the purpose of post-translational processing. Now let's talk about gene expression regulation. Specifically, we're going to start with prokaryotes, and then we're going to move on to eukaryotes. This part of the slide deck is a bit tricky, especially prokaryotic gene expression regulation, so please pay attention. We'll go over these bullet points and then we'll explain with these diagrams that'll really nail down these concepts. So an operon is a group of genes, so it's just a group of genes that come in sequence, and they're regulated by a single promoter. So recall the promoter site is where RNA polymerase is gonna bind so that it eventually can start the process of transcription. Well, it turns out that these genes will be all regulated by the same promoter. Operons are common in prokaryotes and allow coordinated expression of functionally related genes. So as we'll see, for example, you have the lac operon involved in the metabolism. So all the enzymes involved in the metabolism of lactose in a bacteria, for example, or for example, the synthesis of tryptophan, that's another operon. So these will be related. So all the enzymes involved in the synthesis of tryptophan will be on the same operon. And the idea there is that if you want to shut it down, it makes sense to shut the synthesis of all these proteins down because, for example, you have enough tryptophan, so you don't need to synthesize more, so you save energy. The lac operon is an inducible operon. Again, we're going to expand on this in the subsequent slide, whereas the TRP operon is a repressible operon. I'll make sure to clarify these over here. So lac operon, an inducible system. Essentially what it means is that the lac operon by default is shut off, and you have to induce it so it can carry out its function. Now let's get into these three different scenarios that you can have. So just to familiarize yourself with the operon, so this would be an operon right here. You have a regulatory region, you have a promoter region, an operator region, and then you have your enzymes that are involved in lactose metabolism. So here you have lac-Z, lac-Y, lac-A. So these will eventually translate into proteins that are involved, or enzymes that are involved in lactose metabolism. So in the very first scenario, you can see that your regulator right here, your repressor, is going to be able to bind to the operator and block RNA polymerase from carrying out its function. So RNA polymerase binds to its promoter, but then it has the repressor in the way, so it can't transcribe these genes. And that makes sense because, as you can see right here, we have glucose present. And if you have a lot of glucose is abundant, and there's actually no lactose, why would you synthesize the enzymes that are involved in the metabolism of lactose? That would be waste of energy and resources. So that's why it is shut off. And this is what we call negative control, because you have that repressor shutting down the process. Now, in order to activate the process, you need an inducer, and that's why we call it an inducible system. Well, what's that inducer going to be? Well, it's a derivative of lactose, and the idea here is that at this point you actually do have lactose present. So you can see you have lactose present over here, or derivatives of lactose, and they will act as the inducer, bind the repressor, and essentially not allow. So now when you have the inducer-repressor complex, it can no longer interact with the operator, and therefore RNA polymerase will be able to carry out its function. However, as you can see right here, you still have glucose present. So you actually don't really need to synthesize that many lactose metabolizing enzymes because you can rely on glucose for your carbon source. Okay, so while you may have some transcription of these genes over here, you won't have too much transcription because you can rely on glucose. Now, what happens when glucose is absent? So you can see here, now you have lactose derivatives, but glucose is completely active. So first of all, the idea of the inducer-repressor complex is still there, so you still have the system running because the repressor is not able to bind the operator and block RNA polymerase. But in addition to that, when glucose is low, that leads to an increase in CAMP. And when CAMP increases, it can interact with this molecule called CAP. This is no CAP, by the way. All right, it binds to CAP, and then CAP will then go and bind to the promoter. And what it's going to do there is going to increase the transcription. So now, because you have no glucose, you really need to rely on lactose for your energy source. So you're gonna want to ramp up transcription. So in other words, this is no transcription at all because you have negative control. Once you've lifted the negative control, you have some transcription. And then finally, when you have positive control, you have a lot of transcription. The positive control is the idea of cap binding to the promoter. All right, now we have TRP, the TRP operon. This is a repressible system. In other words, by default, this is running. So it's the opposite of lac operon. This one is running by default, and you need something to stop it or to repress it. So essentially what's going on, very similar in terms of the sequence of the operon over here. But what you see is that the repressor alone cannot go and bind the operator and block RNA polymerase's activity. And that's why when the repressor is there by itself, RNA polymerase is still carrying out its function. And only after the co-repressor, which in this case would be tryptophan, binds to that repressor that you can go bind the operator and shut down the process. But essentially when tryptophan is or TRP is at low concentrations, you have RNA polymerase running and generating these, or eventually allowing for the translation of TRPE, TRPD, these enzymes that are involved in the synthesis of tryptophan. And this makes sense since you don't have any tryptophan around and you want to synthesize tryptophan. However, what happens when you get an abundance of tryptophan, so you've synthesized a lot of tryptophan, well, it doesn't make sense to keep on synthesizing tryptophan indefinitely. So at some point what happens is when the tryptophan concentrations rise to a certain level is that the co-receptor, in this case it's tryptophan, will bind to the repressor and then they will bind the operator system, thus blocking RNA polymerase from carrying out its function. And that is why it is a repressible system, because initially it was running and then you had to add some new molecule to be able to repress it, okay, which is tryptophan in this case. All right, so this is just a summary of operon regulation. You can pause and really think about these, but I'll run through them. So lactose absent, repressor binds operator and transcription is off. Lactose present with high concentrations of glucose, repressor is removed, but cap is inactive, so you have low transcription because you can still rely on glucose. Whereas when lactose is present and there's no glucose, you really have to rely on lactose, so the repressor is removed and you have that positive control with cap to increase the transcription. And then for TRP, it's essentially the opposite of that. When you have low tryptophan, it is the repressor is inactive and transcription is on. Once you've synthesized enough tryptophan, now tryptophan acts as the co-repressor and shuts down the process. Okay, now let's talk about eukaryotic gene regulation. And as I mentioned, this is a bit more straightforward than prokaryotic gene regulation. So in the case of eukaryotic gene regulation, you're going to rely on these transcription factors. Transcription factors, you can think of them as binding on one end to a DNA sequence and on the other end like recruiting RNA polymerase to come and bind that and transcribe that specific gene. Now, as I mentioned here, like RNA polymerase is recruited by these transcription factors. And then we have something called enhancers, and I show them over here. This is a small image, but I have it enlarged over here. So what's going on here is you really want to make sure that you understand that enhancers are actually a sequence of DNA. So they're not proteins. They are a sequence of DNA to which transcription factors will bind and recruit a separate part of the genome that's going to be transcribed so they can increase the transcription of that specific gene. And you can see that the enhancer is sometimes very far away from the gene of interest. So this is the gene of interest. This is its promoter. The promoter, recall from the previous slide deck, we mentioned it's at like location like -25, that TATA box. Location -25 just means that it's 25 bases upstream of where you're going to start transcription. So recall the beginning of transcription is going to be +1. Before that is going to be negative numbers. The enhancer can be as far as like -1,000, so like 1,000 base pairs away from that from the beginning of the transcription site. And essentially what's happening is that enhancer region binds to transcription factors, and these transcription factors can include things like steroid hormone receptors. So recall, steroid hormone receptors are involved in the regulation of transcription, whereas peptide hormones, for example, are more short-term effects, like opening of an ion channel, for example. Steroid hormones, such as whether it's estrogen or cortisol, have prolonged effects, and they act directly at the level of gene expression. So they can bind to these enhancers and increase the transcription of a given gene. You can have multiple transcription factors act at the same gene to really increase the expression of that gene. So it could be like for example, a steroid hormone, but also a signaling molecule that interacts with another protein, which is going to act as a transcription factor at these enhancers. And specifically, we refer to them as activator proteins when they're interacting with these enhancers. So like, as I mentioned here, hormone receptors and signaling molecule binding proteins. All right, now let's move on to gene amplification. So the idea behind the way we amplify gene expression in the case of enhancers is by bringing those transcription factors and recruiting RNA polymerase to essentially transcribe a gene or copies of gene that already exist. So maybe like 1 or 2 copies that already exist. However, when it comes to gene duplication, essentially what's going on is that you're not just transcribing one gene. What's going to happen is it's going to make multiple copies of that same gene. So instead of having one of that gene, you're going to have like 10 identical copies of that gene. So now you can really amplify the number of messenger RNAs that you get from that gene, the transcribed messenger RNAs, and then the overall number of proteins that you eventually are able to translate. And one thing I did want to point out over here, that notice because the enhancer is like so far away from the promoter, you have to sort of like loop it kind of in a hairpin loop and bring it closer together like that. So that's just something that they'll sometimes mention on the MCAT. And then gene amplification is commonly seen in rapidly growing cells and in cancerous cells. Here we have a slide on chromatin remodeling and gene expression. I'm not going to go into too much depth with this slide because I do have it on our slide deck on DNA structure and organization, but we'll just briefly mention it here because it is relevant to the topic. So euchromatin is loosely packed and it's transcriptionally active, so available for transcription, whereas heterochromatin is not available for transcription. Histone acetylation generally increases transcription, so you can think of the process of acetylation as loosening up, and this is kind of an oversimplification, but just loosening up that chromatin and putting an open sign on it so that it's available for transcription, whereas methylation is the opposite of that process, where if you were to methylate something, then you are decreasing its transcription. And don't get it mixed up. Like if you see deacetylation, that is the opposite of acetylation. So acetylation activates, deacetylation inhibits. Methylation inhibits, demethylation activates. Now the idea behind the loosely packed and the densely packed chromosome, so if it's loosely packed, RNA polymerase and other transcription factors can come to the site of transcription to carry out transcription. Whereas if it's densely packed, then they're not going to fit there and they're not going to transcribe. And again, you can, if you want more details, more diagrams related to this topic, definitely check out our video on DNA structure and organization. Finally, let's talk about regulatory RNAs. Not extremely high yield, but fair game on the MCAT. So you have everything we've talked about thus far regulates the process of transcription at the level of the DNA. So it regulates the eventual amount of proteins or the presence of a protein that you will eventually get, but it starts very early on in the process from the level of gene transcription from the DNA. However, what happens once you've already synthesized your mRNA transcripts? Well, now these are going to be translated into proteins. So it turns out that there are ways where you can regulate the translation of proteins at this level. And the way that works is with microRNAs, which bind to complementary mRNA. So this is mRNA that you previously maybe needed to synthesize certain proteins, to translate certain proteins. Well, now this microRNA can then bind to those mRNA strands and inhibit their translation, or even label them for degradation. Likewise, small interfering RNAs kind of rely on the same concept and promote the degradation of mRNA strands that are no longer needed. And then finally, small nuclear RNA is a component of the spliceosome, which we talked about in depth in the previous slide deck, so you can refer back to that one. All right, so these are some knowledge checks. I'm just gonna power through. You can pause the video to try to answer them. If you have any questions, clarifications, you can reach out to me and I'll provide you with explanations. All right, here's another one. Okay, and finally, there you go. All right, so that was a dense topic. Genetics is a fun topic to learn and a fun topic to teach, but admittedly, it is a bit of a complex topic. So if you feel like you have any gaps, definitely go watch the video again, or if you have to watch any of the other previous videos related to RNA or DNA, check them out again. If you have any questions, reach out to me. Check out our slide decks and annotate them if that helps you. And also check out our Anki cards, and good luck, you got this.